Hello there and welcome back. So we are in our fourth video series for chapter 1. We're in 1.4 Molecules of Life Proteins. Okay, so as you can see here, there's a lot of meats. I'm sorry to any vegetarians. Okay, so let us look at the learning outcomes for this subtopic. By the end of the lessons, you should be able to describe basic structure of amino acids and how they are grouped. You should also be able to describe the primary, secondary, tertiary, quaternary structures of proteins and the types of bonds involved. You should be able to describe the effect of pH and temperature on the structure of a protein, explain formation and breakdown of dipeptide, and last but not least, classify proteins according to their structure and composition. Okay, so first things first, proteins. They are a very important component in living things. It serves various functions such as defense, transport, and also movement. A protein is a molecule which is made up of polypeptide chains, which is a polymer of amino acids. So this tells us that the monomer or the basic building block of a protein is amino acid. Let's look at the structure of what is amino acid. So ini adalah amino acid. Okay, so what you see on the left here circled N single bond H, that is an amino group. It is basic property. The opposite side, C double bond O, single bond H, that is a carboxyl group. So this is acidic. There, what is in the middle here is a carbon atom, also called an alpha carbon. It is kind of the middle point where all these other groups will attach to. Up here you have the R group, or also called the side chain, and it will determine the classes of each amino acid. R group dia mirip juga dengan R group yang kita cakap dalam fatty acids tadi. Basically, if you have something there but you don't want to describe it in detail, you can replace it with R. Okay, and then you have one hydrogen carbon over here attached to the alpha carbon. So this is basic structure of amino acid. Paling senang saya ingat adalah dia adalah NCC. Okay, so if you see them in a long chain, dia NCC, dia satu unit amino acid. Okay, so we say that we can divide them into groups based on their side chains or R groups, and this is the 4 groups. Kita ada non-polar amino acids. So these are groups that end in CH2, CH3. Polar amino acids, groups that contain oxygen or hydroxyl, which is OH. Thirdly, we have acidic amino groups. This is if the R group is negatively charged. And basic amino acid groups, which is if the R group is positively charged. Jadi kamu boleh tengok sini, pelan-pelan tengok, ataupun Google saja. If you Google, you will be able to find maybe bigger tables with more information and maybe clearer resolutions as well. So go ahead and explore that if you want to. And then we'll go into the next part of the lesson, which is the levels of protein structure. Proteins can have primary level structure, di mana dia satu baris amino acid. It can have a secondary structure, where it folds into this or into this. Later we will see it one by one. It can be tertiary structure. It can be quaternary structure, where you have more than one polypeptide. Okay, so let's look at each of these structures one by one. The primary structure, kadang kita tulis satu and then the degree punya symbol. So this is the primary structure. It is when you have amino acid sequence. It consists of the amino acid sequence of a protein. The only bonds in here are peptide bonds, and a slight change in the primary structure of a protein can affect the whole conformation and ability to function. So sini kamu nampak dia panjang berantai. It's like a really long chain. If you look at the chain, this is what's inside. Ini yang saya cakap tadi, one amino acid is NCC. Ini amino group, carboxyl group dia sudah donate dan form a peptide bond with the next amino acid. This is an example of a primary structure with only 3 amino acids. Kalau kamu ada banyak lagi amino acids, banyak lagi lah di sini tengah bahagian purple nih. Okay, the green line is showing the peptide bond. Okay, so dia satu rantai panjang saja, nothing special. We will go on to the secondary structure. So if you have that rantai panjang, and then you fold it, or you coil it, you will get the secondary structure. Okay, this is what is meant by a secondary structure with an alpha helix or a beta pleated sheet. So if this is your primary structure, kalau primary structure kamu adalah satu keping kertas yang flat macam ni, secondary structure is this. I am going to coil it into an alpha helix. Okay. When I coil it like this, dia macam ada pusing begitu kan? This is your alpha helix. If I had a longer paper, it would look nicer. If I want to fold it into a beta pleated sheet, this is how it starts. Saya lipat, 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 lipat dia. Lipat lagi. Jangan ketawa saya lipat lambat. Okay. And then you will have this. Okay. When you have this is your beta pleated sheet. That is what is happening over here. What is making sure that these things stay together are hydrogen bonds. So let us have a look at the slides once again. So here you can see secondary structure is a result from hydrogen bonds at regular intervals along the polypeptide backbone. Typical shapes are the alpha helix, like I said earlier, and the folded or beta pleated sheets. Structural properties of silk are due to beta pleated sheets. So kain sutra, silk, it feels that nice is because it has this structure. And the presence of hydrogen bonds make each silk fiber stronger than steel. So kain sutra tu walaupun dia nampak macam lembut, senang mapegang, tapi dia actually sangat tahan, very durable, strong, because of these hydrogen bonds. And yes, silk is a type of protein. Okay, so sini saya ada bulatkan mana yang ada hydrogen bond. All these dotted lines are also hydrogen bond. We are moving into the third level or the tertiary structure protein. So after you have the beta pleated sheet and the alpha helix, your very long polypeptide can also pusing dan belit dan tergumpal. Okay, it can coil, it can bundle up, and basically fold into its own unique shape. How it gets to the tertiary structure is determined by a variety of interactions among the R groups and between the R groups and the polypeptide backbone. What do we mean by these interactions? Interactions means it can be bonds, it can be hydrophobic interaction. So kita tengok sini, ada contoh dia. Example is hydrogen bond. Another example is ionic bond. Third is hydrophobic interaction. Fourth is van der Waals interaction. And fifth is disulfide bridges. Take your time, read these notes. If you need to ask me question about apa benda ni miss saya na paham, feel free to ask me in the Telegram group. Okay, last but not least, we are talking about quaternary structure. So quaternary structure, dia tiada sudah special bond lain. Dia basically if you put more than one polypeptide together, that is quaternary structure. Okay, so you have aggregation of 2 or more polypeptide subunits. One example of quaternary structure protein is collagen. Collagen is a fibrous protein of 3 polypeptides. They are supercoiled like a rope, and this provides the structural strength for their role in connective tissue, and then they make your skin look very young. Another example of quaternary structure protein is hemoglobin. Hemoglobin is a globular protein, so globular means bulat-bulat, okay? globular protein with 2 copies of 2 kinds of polypeptides. So dalam hemoglobin, we have 2 kinds of polypeptides, which is alpha chain and beta chain. Alright, once you have a change in the amino acid sequence, okay, kalau kita tertukar pun satu part dia itu, ada kemungkinan protein kita tidak akan berfungsi dengan baik. Okay, if you change even one amino acid, there's a chance your whole protein may malfunction. So let's take the example of sickle cell disease. Okay, it is when you have a change in the primary structure of that protein. In an individual with sickle cell disease, you have abnormal hemoglobin proteins developed because of a single amino acid substitution. So di sini, you see it's supposed to be glue, and then it gets substituted with val. All right. And yeah, so that's just to show that every amino acid is important. Okay, we are moving on to the next part of proteins. Next, you will learn something that you probably learned in school before. We are talking about the effect of pH and temperature on the structure of protein. So the protein's conformation or shape can change in response to the physical and chemical conditions of its surrounding. Some proteins can return to their functional shape after denaturation, but most of them cannot. Denature basically means dia terlari daripada bentuk yang sepatutnya. Denature means it is not its normal protein conformation, not its normal protein shape. Factors that can denature a protein are extreme pH and high temperature. Kalau pH untuk protein itu adalah 7, if the normal pH for it is at 7, and you put it into a solution that is pH 9 or pH 5, it is going to denature. If that protein is usually at 37 degrees Celsius, human body temperature, you heat it up to 50 degrees Celsius, it can denature. Both these factors will disrupt the hydrogen bonds, ionic bonds, and disulfide bridges that maintain the protein's shape. So those bonds that are usually involved, secondary and tertiary, and also quaternary, I think, level of the protein. Dia akan terkacau dan dia akan terurai. Okay. Next one, we are going to learn about how dipeptides form and break down. Ini mirip juga dengan macam mana saya ajar yang formation of disaccharide itu. So we start with amino acids, 2 amino acids. This one we are going to focus on the carboxyl group, and on the right we are focusing on its amino group. We do not need to focus on the side chain. So for this example, we just replace it with R. Sebab kita tidak pentingkan dia sekarang. Okay. So if you want to form a dipeptide, you take 2 amino acids, you remove 1 molecule of water. This process is now called condensation, and you will have a dipeptide with a peptide bond. If you want to break down the dipeptide, kita lebih kurang kasih terbalik sejak itu gambar aja. We have the dipeptide here, label the peptide bond, plus H2O, lukis anak panah, tulis hydrolysis, and then you draw the original amino acids. Kalau di sini soalan bagi R, kamu tulis balik R. Kalau soalan bagi CH3, kamu tulis balik CH3. Okay, so you'll have the chance to practice with this later. Let's talk more about how proteins are formed. So proteins or polymers of proteins is formed from condensation of amino acids, which are joined by peptide linkages or bonds. I already talked about this before. I showed it to you with the diagram with the green line. Okay, ini lebih kurang macam mana dia lah. The backbone, if we talk about protein backbone, it is the NCC, NCC, NCC. The side chain is the R. R side chain, apa yang berjuntai keluar, itu adalah side chain. Okay, moving on to the next segment. In this next segment, we are talking about the classification of protein based on their structure or shape. We can classify proteins into three main groups. The first is conjugated protein, which is a compound made up of a protein molecule and a non-protein prosthetic group. The second is globular proteins. Globular proteins have complex tertiary and sometimes quaternary structures. They can be folded into spherical or globular shapes. Globular proteins are usually water soluble because the hydrophobic side chain is inside, not outside. So yang outside is the hydrophilic ones or normal, non-charged. Okay, and then they have roles in metabolic reactions. Examples of globular proteins are enzymes and hemoglobin in blood. Third classification of protein is fibrous or fibrous proteins. So fibrous proteins have little or no tertiary structure. They're usually secondary. They have long parallel polypeptide chains. They have cross linkages at intervals forming long fibers or sheets, and they are usually insoluble. Many of them have structural roles. So examples of fibrous proteins are keratin in hair and the outer layer of your skin, collagen. So let us look at the importance of protein. First things first, it is involved in nutrition. So proteins such as digestive enzymes, an example is amylase and lipase, are involved in food digestion. Second importance, proteins are involved in transport of respiratory gases. An example is hemoglobin, which transports oxygen. The third is proteins are involved in our body's immunity. Antibodies will recognize the antigen and defend the body against diseases. Number four, proteins are involved in coordination. Proteins can be hormones, and those hormones can act as chemical messengers. Such examples are insulin and glucagon, which regulate our blood sugar level and controls blood pressure. Next, proteins are important for growth. We have hormones that control growth and metabolism, such as thyroxine and growth hormone. And proteins can also provide us with support and aid our movement. Examples of support is keratin that you can find in your hair, keratin on the surface of your skin, collagen in your tendon, collagen in your ligaments, collagen in your arteries, and these provide you with the support and strength. You also have contractile proteins such as actin and myosin in your muscle cells, which are involved in muscle contraction. Okay, that is it for proteins. I hope this video has been helpful rather than confusing. Let us have a quick run through. Okay, kita ingat balik apa yang kita sudah belajar tadi. So proteins are made up of amino acids, which can be divided into four groups. You have several levels of protein structure: primary, secondary, tertiary, quaternary. Proteins can be denatured by extreme pH and high temperature. You can draw and show formation and breakdown of dipeptides. Proteins can be classified into three main groups. And we also went through the importance of proteins. These are the learning outcomes. I hope you can reach them. And what you should do now is you should once again take out a piece of paper or your notebook. Please try to draw the basic structure of an amino acid. Label whichever groups that you know how to label. Second is draw the formation of dipeptide. So ada slide tadi yang menunjukkan macam mana kita boleh membentuk dipeptide, how you can form dipeptide through condensation. Draw that diagram for me, please. And number 3, in a table, please list out the possible bonds that can be found in a primary, secondary, tertiary, and quaternary structure proteins. So, lukis satu table. Dalam table tu kamu tulis primary, apa bond yang ada? Peptide bond. Secondary, peptide bond, hydrogen bond. tertiary, and quaternary. Begitu. And then upload your answers in the Google Classroom. That's all for this video. I'll see you again in the next one. Thank you and see you again. Bye!